Okay, let's take a look at some general principles of alternate splicing. Now this is just like you get cell-specific changes in transcription of a given gene that require specific transcription factors. Here's where the specific splicing factors come into play. Specific splicing factors, they're going to be found only in certain cell types, and they will govern splicing of particular genes, or rather particular pre-messenger RNAs, and not of others. And different cells are going to have different complements of specific splicing factors in there. Now let's take a look once again at these intron-exon boundaries. Okay, here's an intron-exon boundary. The AG over here and the GU over here, those are absolutely invariant. They're absolutely essential. Now, things like U1 and stuff like that, U2 binds down over here, that's going to vary depending on what else we have, what other sequences we have nearby. These sequences, you've got to have them for sure, but what else is nearby is going to govern how tightly these various things bind to the splice sites and other things like that. So you can have, for instance, what we could call a strong splice site. Just like we can have strong promoters. And a strong splice site is a site that spliceosomes will preferentially assemble to, because they'll bind the appropriate components tightly. In fact, what I'm going to do, and this is going to be a crude spliceosome, is I'm just going to make this thing. Okay, so this is a strong splice site. Now we have wimpy, weak splice sites. Weak splice sites generally won't bind the components very strong. Okay, and weak splice sites. Weak splice sites bind the spliceosome components very weakly. So for the most part, they're going to fall off before you can get the rest of the spliceosome coming together. Does this sound very similar to what we see with the basal complex? Okay, yeah, it's the same basic principle here. Some intron-exon boundaries are going to promote spliceosome assembly, others won't. So in this case here, with a weak splice site, no spliceosome. Okay, over the strong splice site, we get splicing. So now we join the two exons together. Over this wimpy site, we don't get splicing there. Now suppose we happen to have, let's just suppose that this is all part of the same gene. We're going to do this, right? Okay. And here's another one, and here's a strong splice site. What would happen in a situation like this? We've got a strong splice site here. This is the same exon, just had to bend it a little bit. We've got wimpy splice sites here, and we've got strong splice sites here. Well, we could form a spliceosome from here to here. we would not form a spliceosome from here to here. So if this is going to be exon 1, this is exon 2, this is exon 3, and this is exon 4. What's going to happen here? We get splicing exon 1 and 2. 3 and 4 are going to get spliced together, but 2 and this portion of 3 are going to get discarded. Okay, so let's see. Those guys get spliced together, and over here, this place is so many forms, so we're going to actually, in this case, we'll have the whole thing put together. But let's suppose, find another situation. Okay, let's make this over here. In this case, we're going to splice all these exons together. In this case here, let's make this a weak splice site. Again.
Well, why did they splice together?
All right, well, we've got these two will be joined together. And, okay.
E2 to E3 wouldn't exist.
Yeah, okay, yeah, E2, yeah, E2. And,
Do you want to erase E2 here?
all right, yeah, all right. There we go, sorry about that. All right. Now we have a weak site here.
Strong there, weak there, strong there.
Okay, we have strong here.
Why would E2 not exist? Wouldn't
Well, let's see, let's
together?
see here.
E1 and E2 do.
Okay, E1 and E2 will be joined together.
Right.
So then there's a space and then
3 and 4.
no.
All right, 3 and 4 will be joined together here. And the question is this one, we're not getting it, oh, that would actually lead to intro, oh, yeah. In this case, actually what we'd have, okay, we'd actually have intron sequences here, which could happen.
That's what I was thinking.
Okay, all right, we finally got that one figured out. All right, in that case. Okay. Now, in this case, you would have, for instance, the intron sequence would actually be the final messenger RNA. It would code for something. That would give you a different version of protein. If we had strong splice sites across the board, we would have, so let's kind of do that. Okay, let's make strong sites here. Let's put strong sites across the board, and that's so you know, okay, strong. Ow, I hate this job. Okay, so spliceosome forms here. I'll just call SPL for spliceosome. Spliceosome forms here. In this case, we're going to get all the exons. 1, 2, 3, 4. Put together. Okay, let's do another version here. Let's suppose we're going to make these two weak. Once again. And this one is going to be weak as well. This will still be strong. Okay, we're not going to be able to make a spliceosome that goes from here to here because it won't assemble very well. We're not going to be able to have a spliceosome go out to here. Instead, what we're going to have to do, or in fact, let's make this one strong. We'll make this a strong one. So we got a strong one here, a weak one here. We're not going to get spliceosomes going from here to here because you can't stick there, or from here to here. But what we can do is get a spliceosome bridging all the way across. And we end up in this case with exon 1, exon 2. Exon 3 is not part of the ball game. Exon 4 is joined to it. This intron and this intron are discarded. So we have like that. So, if you have a default situation, depending on what splice sites are strong and what ones are weak, you can have a default situation where you're going to discard certain exons. Or in certain cases, you may actually keep introns in the final product. Question?
Is it, would it be possible for like, say, the spliceosome to attach to a leak, but then fall off after it has started? the
Well,
splicing?
yeah, that could, and that would be a random thing, so you would get some mis-splicing there. And here again, just like the thing that you can occasionally get a basocomplex forming even at weak promoters, it's not likely, but you might get some. So you will get occasional mis-splicing. And here again, there will be mechanisms often to detect that and then just destroy it before it ever goes out of the cytoplasm. But you're right on that. You could get, since once again, it's a random kind of process, it's more likely spliceosome to form and hold together long enough at a strong site than a weak site. But once in a while you get lucky or perhaps unlucky. So that's a good point there. Okay, so by different combinations of strong and weak splice sites, we can end up with different alternate splicing patterns. Now what we're going to do here is this can be regulated by specific splicing factors. Okay, what we're going to do is make 3 exons and 2 introns. So exon 1, E2, and exon 3. Okay, let's suppose all of these things have strong splice sites. So what's going to happen if we have spliceosomes bridging here to here and here to here? So in this case, the default situation is we got all 3 exons put together. Okay. Now, let's suppose that another cell type has a specific splicing factor. That protein is going to bind very tightly to this particular boundary and to this boundary here. In this case, these strong splice sites, now we're going to have these splicing factors, and what they will do is they will bind to the same sequences that the appropriate parts of the spliceosome bind to. So they bind to it like here. And what that means is they're going to prevent a spliceosome from assembling because the components that have to assemble here aren't anywhere to be had. They can't displace these splicing factors. So in this case, we've blocked these 2 splice sites. We have splicing inhibitors. And now what we'll have is a spliceosome forming all the way from here to here. And the end result, because we blocked these 2 splice sites, the end result here are we're going to join exon 1 and 3 together. And meanwhile, these 2 intron sequences plus exon 2 is discarded. And all we needed in that was a single specific splicing factor that blocked spliceosome formation at these 2 junctions. And that caused us to get rid of that entire exon. So in this case, a strong splice site, but some splicing factor is regulated. And let's put this down.
So it could happen as in like E1, E2, E3, but it could cut E2 out, or it always is going to cut.
Well, in this case, you would have, since you can't get a spliceosome forming from here to here, this is a strong site. This is a strong site. These sites, which used to be strong, are now blocked. So spliceosomes can't form here and here. So instead, we've got a bridge all the way from here to here, because we can't form one over there.
And what blocks it?
Okay, a specific splicing factor. In other words, a protein that binds the same, or at least close to the same sequences that the appropriate, like U1 or whatever the case is, would bind to. It binds to it tighter than U1. So U1, for instance, in this case, well, using this one as an example, ordinarily U1 would bind here, right?
Right.
Okay, now the splicing factor's there. It grabs onto that splice site so tightly that U1 can't push it off and bind to it. So without U1 forming there, okay, well, you're not getting a spliceosome. But it could form elsewhere.
Okay.
Okay, so now we've got that. So in this case, what we've done is we tossed out an exon simply by putting the appropriate, like what's effectively a blocker of spliceosome formation at a specific spot. So strong splice sites are regulated. We could call them by splicing inhibitors. Okay. That's one example where we have a strong site, and to regulate it, we have to prevent spliceosomes forming at that site. We have to block it. Yes?
Are all specific splicing factors inhibitors?
No, because I'm about to go to the opposite thing. Now, let's suppose we're going to change things here.
Is that accelerated?
Accelerate. No, regulate. Regulate. Okay, we're going to change things here. Okay, that last example, all these splice junctions happen to be strong junctions. Now, let's change things. Let's say that this is strong, this is weak, this is weak, and this is strong. Okay, in this case, the normal default situation, since these are weak splice sites, spliceosomes, they'll start forming here, but they're not likely to stick here or here very well. So in this case, generally what's going to happen is the spliceosome will bridge E1 and E3. Here's your spliceosome. So ordinarily what we will get is what we saw before, exon 1 and exon 3, but exon 2 and the 2 intron sequences are discarded. So that's our default situation. Now let's regulate things. What we're going to do now is we're going to attach, at these two junctions, we're going to attach some more specific splicing factors. In this case, these splicing factors are going to bind to the sequence tightly, and they will bind to the appropriate spliceosome component. So now we have some splicing factors. We could call them splicing activators. And what we've effectively done is we've turned weak splice sites into strong ones. So now what happens is spliceosomes assemble. And in this case, all 3 exons get joined together. So, one cell type. We don't have any specific splicing factors for that RNA. Ordinarily, what we will have is just exon 1 and 3 joined together. But a different cell type happens to have this one particular splicing factor that promotes spliceosome assembly where it binds. Now we can put 1, 2, and 3 together in that cell type. Now imagine the complexity if you happen to have 20 or 30 introns and exons. You can have each cell type having its own complement of splicing factors and choosing what introns and what exons are going to be joined together.
And this is what gives you all those options.
Right. It's the splicing factors that kind of make the choice of what's getting spliced and what's not.
Are there names for these, like?
For some of them, I think we've identified, I think they've characterized a few handfuls of specific splicing factors, and they'll give them different kinds of names. There's probably a whole slew of them that we don't know. Don't know many offhand.
But we don't want to know.
No. But there are a limited number that they've actually characterized and stuff in detail. But a lot of them, we know they're there, but we haven't really looked at what the details of. That would be cool. You know what would really be neat is look at this thing at the whole molecular, tertiary, quaternary structure level. That would be cool. But that's come back in a few decades and maybe we'll have that. Okay, well, So in this case, weak splice sites can be regulated by what are effectively splicing activators. Okay. So in this way, we can do things like discard exons. In some cases, include whole introns into the final messenger RNA. And those introns may actually code for something and you get a different version of a protein. In that case, the protein is probably going to be quite a bit different from the default protein. It may even have a different function, but you can do that. You can include introns into there if you want to. And there's one other little thing. We can even decide to keep part of an exon, or maybe even part of an intron in some cases, usually part of an exon, and throw another part out. So we're not limited to getting rid of or keeping whole exons. We can sometimes get rid of parts of exons. And the way we're going to do this is, this actually came up when they first discovered splicing. What people did was they started playing around and start doing things like, okay, this AG on one end, this GU on the other end. Let's start seeing what happens. We always see this in all intron-exon boundaries. Let's see what happens if we change that. So they changed that, and normally that would just prevent splicing entirely. But not always. Sometimes you would get some splicing occurring, but it would occur at a somewhat different spot than the original location. So we had something like this. Here's an exon. Here's the intron. Okay. All right, let's change this sequence. We're going to change it to GA. Well, end result, most of the time, it's no splicing. Okay? All right, but sometimes what might happen is you would find that there was a second splice site located somewhere else, often inside an exon. And because ordinarily, if you have the sequences intact here, this is where you get splice, you join the two exons together and throw the whole intron out. But sometimes there are these hidden sites. You didn't know they were there unless you blocked, by changing the sequence, the original splice site. So they call these cryptic, hidden in other words, splice sites. And what would happen is if you block that, you would get a spliceosome going from here to here. End result would be you would discard this part of exon 2, keep this part. Sometimes they would find these cryptic sites located in introns. Now they were cryptic, in other words, they were hidden because ordinarily the normal splice site would go. But only those cryptic sites would only be active if the original splice site was damaged in some way. Now the question is, why would you have those things? Now most RNAs don't, but some do. So what are they there for? Are they there as a backup in case some crazy molecular biologist decides to play around with critical sequences at the end of junctions? Nature's way of protecting cells from crazy molecular biologists and doing genetic engineering experiments? No, probably not. That can help. But instead of a crazy molecular biologist changing the sequence, suppose we had a splicing inhibitor here that we find in one cell but not the other. Ordinarily, most cells, you would throw the entire intron out and keep both exons and glue them together. But one cell type happens to have a splicing factor that blocks this sequence here, and lo and behold, you've got that cryptic splice site. So now we're going to get a different version of the final RNA with parts of exon 2 missing. Or if we happen to have a cryptic site located inside an intron, we would actually include intron sequences into this final messenger RNA. So we could do those kinds of things. So, the idea of these so-called cryptic splice sites allow us not only to select the idea of keep what exons and introns to keep and what to discard, but also we can use that to select and discard parts of exons, or maybe include parts of intron sequences into a given messenger RNA.
But
So,
in both of those examples, it
Uh-huh.
kept parts of the exon 2, right?
yeah,
And which one it kept
yeah.
one?
It's we cut all the way up to that cryptic site. That part would be, well, let's put this guy in here again. Here's exon 2. Here's our cryptic site. Okay, well, ordinarily what's going to happen is we're going to take all of exon 1 and 2 and join it together. But if we block this site, we use the cryptic site instead, and the end result is we're going to discard that and part of exon 2, but not the whole thing.
Just need to get a functioning protein.
Yeah, its function, it may be spelled cell-specific version, or it may have a different effect than the original version. In fact, we're going to see that in just a few minutes. So what we have here, looking at splicing, we have several levels of, oh boy, don't have much time either. We have several levels of complexity. Alternate splicing, by making strong splice, specific strong splice sites weak, or specific weak splice sites strong, we can select and pick and choose what exons to keep, what exons to discard, and sometimes even throw in introns into the final messenger RNA if we desire to do so. But we can be even a little more finesse, because if we have cryptic sites, in other words, that are ordinarily not active unless the regular traditional site is blocked, then we can do things like discard parts of an exon or keep parts of an intron. So that gives us additional levels of complexity to make different kinds of proteins from the same initial RNA. So that's pretty cool. That gives us a lot of layers of complexity. And here again, these different types of proteins, these different splicing factors, different cell types. So let's put this down. And these splicing, so different cell types may have different, will have different complements of splicing factors. And the other thing is that certain cell types, certain splicing factors, you may be able to regulate them. Maybe the same cell that under certain circumstances will want to splice a given RNA one way, under different set of circumstances, they may want to splice the same RNA another way, in the same cell type at the same time, depending on environmental cues, what it needs to do. Well, if you have a regulated splicing factor, where you can turn it on or turn it off at will by phosphorylation, whatever you want to do with it, you can have that level of control. So a cell at a given moment can switch from one splicing pattern to another to meet whatever its environmental challenges are. So that's a cool thing too. So we have a lot of complexity, a lot of neat things with splicing and splicing factors.